Hi, everybody. This is Matt. I am from Seattle, Washington. Next month, I'll be a 10-year thyroid cancer survivor. And you are attending session 185, Thyroid Cancer Trials 2022, with Dr. Bruce Robinson, endocrinologist in Australia. Dr. Bruce Robinson is an endocrinologist and professor at the University of Sydney in Sydney, Australia. He has been the head of the Cancer Genetics Unit at the Colling Institute of Medical Research at the Royal North Shore Hospital since 1989. He has been chairman Hawk My Foundation, a major program in medical and health education and exchange with Vietnam since 2001. In 2015, Dr. Robinson was appointed as chair of Australia's peak advisory and funding body for medical research, National Health and Medical Research Council. His research is focused on identifying genetic changes which either predispose or directly cause endocrine tumors, and among other highlights has been the formation of an international consortium of families from around the world to study medullary thyroid carcinoma and pheochromocytoma. And I can never remember how to pronounce that. And with that,
you did well.
Welcome, Dr. Robinson.
Okay, well, look, good morning to everyone here in Australia, and good afternoon and good evening to those in America. Thank you very much for asking me again, Gary, to talk to our colleagues here in Australia about the current status of clinical trials. As you can see, I'm sitting in my car. I'm able to talk to you this morning for about 40 minutes. I'm afraid I can't do the whole hour, but I'll talk to you about some of the progress that's been made in thyroidology generally, particularly thyroid cancer. And I'll also talk to you about the current status of the clinical trials that are open here in Australia. First of all, it's fair to say that despite COVID and the last 2 years, clinical trials in thyroid cancer have not really slowed down at all. It's been quite astonishing that unfortunately the need for these clinical trials has persisted, and at the same time, the companies have been very keen to see recruitment continue. There's also been a lot of progress that I just mentioned briefly in understanding the molecular genetics of thyroid cancers. And in Australia now, it's becoming much more commonplace for patients who have radioactive iodine refractory thyroid cancer to have genetic testing done on their tumor tissue to try to identify potentially actionable mutations, or that is mutations that would respond to treatment with any of a number of different drugs that have been developed now. That testing is available in most capital cities in Australia, certainly in Sydney and in Melbourne. There are very good groups that are able to provide that genetic testing. And I'm also pleased to say that our pathologists here in Australia have really led the way in helping to try to redefine some of the criteria for diagnosing and staging various forms of thyroid cancer. So the highlights of the last year from a research point of view, from my perspective, have been the publication of a couple of trials, but also another really important highlight has been the publication by Professor Anthony Gill, who's a pathologist at our hospital here in Sydney, the Royal North Shore Hospital, of a new staging system for medullary thyroid carcinoma. Some of you who are online, I know, will be interested in medullary thyroid cancer. That has recently been accepted in the journal Thyroid, the Journal of the American Thyroid Association, and already is being referenced by people around the world. Anthony Gill is a particularly high quality thyroid pathologist, and we're very, very fortunate to have him with us. In fact, he's so talented that even by the age of 40, he'd won a prize in America from the American College of Pathologists for producing the best paper by a pathologist who was aged under 40. So that's been a real highlight for us. So too has been the ongoing work that I've been involved in with the American Thyroid Association in the revision of the American Thyroid Association's cancer guidelines. Gary has been involved in that. Unfortunately, the Americans seem to schedule these calls generally at about 3 o'clock in the morning for me, but they've actually become a bit better and schedule them now at 6 or 7 a.m. So it's at least a little bit more palatable time to wake up to contribute to those sessions. Those guidelines have a few changes in them, nothing dramatic, but I can assure all of you out there that there has been a massive amount of time and effort put into these guidelines, particularly by our colleagues in America, particularly Matt Ringel from Ohio and Julianne Sosa from San Francisco. Both of those people, but a lot of other people too, have put a massive amount of time and effort into updating these guidelines based upon the literature as it's been published in the last 6 or 7 years. So what's actually been completed and what is currently ongoing as far as clinical trials in thyroid cancer is concerned? Well, the first thing is that the study of, now I'm talking about medullary thyroid cancer, the study of Loxo's drug, now owned by Eli Lilly, called Loxo 292, now renamed selpercatinib, was completed and published. And selpercatinib is now a drug which is available to us for clinical trials in Australia. Having completed the first trial, there is now a second trial that's open in Sydney, Melbourne, Brisbane, and Perth. As far as I know, I'm not so sure about Adelaide, but that trial, which is on the back of the trial that showed that selpercatinib had effectiveness in patients with medullary thyroid carcinoma who have a RET codon 918 gene mutation.
Looks like we have lost Dr. Robinson. We'll give it a second for him to reconnect.
Sending me a message, Matt, that it was overheating, so I've stood outside in the breeze. So can everyone hear me now?
Yeah.
Okay, so look, I'm sorry about that. What I was in the process of saying is that the first study, which examined the utility of selpercatinib in medullary thyroid carcinoma, was published last year, and that was a positive study showing this drug certainly has efficacy in patients with medullary thyroid carcinoma who have a codon 918 mutation. So a very important part, as I touched on earlier, a very important thing that to have done if patients have medullary thyroid carcinoma, I think at the time of diagnosis, is to have their RET codon 918 mutation status assessed, because it's often difficult 10 years down the track to find the piece of tumor that might be required to undertake that sort of analysis. So on the basis of that study, Eli Lilly, who now own selpercatinib, decided to set up a second study comparing cabozantinib or vandetanib with selpercatinib to see which of these might be superior. Obviously, they were anticipating that selpercatinib will be the superior drug. That study, which is known as the JZJB study, is currently recruiting patients in Sydney, Melbourne, Brisbane, and Perth. The only hiccup with the study has been that initially, whilst patients are randomized 2-to-1 to receive selpercatinib instead of cabozantinib or vandetanib, that vandetanib supply globally is unavailable, and so the study is now a comparison of selpercatinib with cabozantinib alone. That's a good study. Obviously, Eli Lilly expect that selpercatinib will prove to be the superior of the two drugs. Otherwise, they wouldn't be paying for this drug, for this study to be undertaken. It's good that patients in Australia have access to this trial through a number of different centres. Sometimes accessing this trial proves to be difficult because there has to be a degree of disease progression demonstrated before one can access the study. And we've had a few people who've not been able to get into the study. Fortunately, also open is an expanded access program, which Eli Lilly run, and patients are generally able to get direct access to the drug through that expanded access program. They do, however, make patients go through the screening process for the trial study, first of all, and they have to fail that before they can get into the expanded access program. And of course, that whilst that might sound harsh, without such rigor and discipline, a study won't be able to be done, and we won't be able to get the evidence that suggests that one is a superior drug to the other for the benefit of patients who might come along behind you. The other study, which is almost open in Australia, is known as the pralsetinib study. Pralsetinib has not been used in Australia previously. The study initially showing that it was effective in patients with medullary thyroid carcinoma was done only in the USA. Again, it proved to be effective with similar levels of effectiveness as selpercatinib. And so that study is now an additional study is being performed comparing selpercatinib with cabozantinib. In this one, the randomization is 1-to-1 rather than 2-to-1 in the selpercatinib study. So you're twice as likely to get selpercatinib in the selpercatinib study as cabozantinib, but equally as likely to get pralsetinib in the pralsetinib study. The problem with the pralsetinib study is that Roche at the moment don't have drug to be able to supply to people. So although technically the study is open, we're not able to recruit anyone into it at the moment. So for medullary thyroid carcinoma, the landscape is quite good for patients in that there are at least 2 trials open, and I'll come back to medullary thyroid carcinoma in a few moments. I'd just like to switch tack now and talk about patients with differentiated thyroid cancer, who patients with differentiated thyroid cancer, traditionally, of course, as you know, are treated with radioactive iodine. And when they become radioactive iodine resistant, they may also be now in Australia through the PBS, the Pharmaceutical Benefit Scheme, be treated with lenvatinib, which was shown in a study published several years ago, led by Martin Schlumberger from Paris, that lenvatinib had very good effectiveness in helping prolong progression-free survival and even overall survival in patients with poorly differentiated radioactive iodine resistant thyroid cancer. Now, there has now been an additional study performed and funded by a company in California called Exelixis, who have another drug called cabozantinib. Cabozantinib has now been shown to be an effective second-line therapy for patients who have failed on treatment with lenvatinib. Now, lenvatinib resistance is now a recognized phenomenon, and eventually people who are on this drug do become resistant to it. And one of the things that we are studying in collaboration with our colleagues at Mass General is the reasons for that resistance to all of these drugs actually becoming a problem. We're doing that collaboratively because we need to have people who've been on the drug, who've then become resistant to it, who've had biopsies of their now resistant tumor, so that we can understand what new mutations have occurred to cause that resistance. And of course, having identified the cause for that resistance, the companies are all interested in this information because they're very keen to start to design drugs which overcome the new mutations that have arisen in these people that have caused their tumors to become resistant. And already with Loxo, the Loxo company, now owned by Eli Lilly, as I said earlier, have already developed an additional drug which can be used in patients with medullary thyroid carcinoma who become resistant to selpercatinib. Now, so cabozantinib is now recognized through a publication that occurred only in the last few months, led by Marcia Brose, but we were involved in that study at North Shore. It's been shown that nearly 70% of people who become resistant to lenvatinib with differentiated thyroid cancer are able to respond to cabozantinib. So I think all of you out there should take heart that should patients develop resistance to any or each of these drugs, that the big pharmaceutical companies and us are actively looking to try to find the mutations which cause the resistance, first of all, and secondly, new drugs that can be used and put into trial to help overcome that resistance. So it is a very exciting time. It's a challenging time. I think we all feel the pressure of time to get this work done. As I've said a number of times, we tend to do this work collaboratively because we need speed. We need significant numbers of patients that we can study, and rarely does any one centre have sufficient patients available to be able to answer all of these questions by itself. Fortunately, the thyroid community, the thyroid cancer community, is a very tight one. Rory Clifton Bly and myself are both members of an organisation known as ITOG, the International Thyroid Oncology Group, which is very active in helping to try to work with pharmaceutical companies to design these trials. And that's a good thing because it means that some of the physicians who do this work are actually in the driver's seat when it comes to working with companies to work out what trials might be done. It's not just all commercially driven. The other big advance in Australia in the last 12 months has been, as I touched on earlier, the much more widespread use of mutation testing on patients' tumours to actually identify the specific mutations that they might have that might make them candidates for some of these drugs. Now, I've talked about trials. What I haven't talked about is what's known as off-label use of drugs. When we undertake that mutation testing in people, we often find somewhat unusual or really quite rare genetic alterations where bits of one chromosome are hooked up to bits of another chromosome, or specific mutations have been identified. And these mutations are not necessarily just confined to thyroid cancer patients. They're present in patients with a number of other tumor types. And so whilst there isn't a massive amount of data on their use in patients with thyroid cancer, globally, there is a significant amount of use with these relatively rare drugs that can be used for relatively rare mutations that can occur in other tumor types such as breast, mouth cancer, melanoma, etc. Exactly the same mutations present in thyroid cancer, one can reasonably extrapolate, might be amenable to treatment with other much more specific drugs. And there are drugs whose names you may recognize: entrectinib, larotrectinib, which have all been used in patients with thyroid cancer off-label. All that one has to do is contact the TGA and you put in what's called a special access scheme request, which in essence means that the physician is taking responsibility, having consented the patient to using this drug, which will be registered in Australia, but which is not on the pharmaceutical benefit scheme in this country. And then the next step is hopefully having a company who owns the drug be willing to provide it to patients either free of charge through a compassionate access program, or alternately through an expanded access program for which there may be a charge. Sometimes those charges are very minimal, sometimes they're quite significant. And in fact, one of the companies charges the best part of $7,000 or $8,000 a month for some of these drugs. And so there has been crowdfunding of some patients to get access to them. But often if you work in a hospital with a good drug committee, then the state government ends up funding it because they will look at a patient's circumstances and often agree to fund it through the state hospital system rather than the federal system, which is essentially the pharmaceutical benefit scheme. To get these drugs onto the PBS is quite a challenge. And unfortunately, the number of people we have in Australia who would need them is relatively small. And so it becomes a commercial decision on the part of the companies that it's just not worth the $150,000, $200,000 that it would cost them to get these drugs through the Pharmaceutical Benefits Advisory Committee and onto the PBS, where, of course, they would cost you the $37 or $38. In fact, I think it's just been brought down to $30 per script for these medications. So that comes back to what I started off talking about this morning. It's really important, I think, that particularly those patients who have radioactive iodine resistant disease, but arguably all patients with thyroid cancer do have genetic testing done on their tumor, because if a patient does have one of these rare genetic alterations, then often the best thing for them is to be treated relatively early with one of these drugs, even rather than them having radioactive iodine, because often patients with these genetic rearrangements don't respond all that well to radioactive iodine treatment. Now, of course, the one that the genetic rearrangement that everyone knows most about is called RET-PTC. This is the one that's quite prevalent in people who've been exposed to radiation, such as around the Chernobyl nuclear reactor accident, Fukushima, etc. People who've been exposed to large amounts of radiation have a higher frequency of these mutations. And in fact, many of these patients with differentiated thyroid cancer are actually amenable to treatment with selpercatinib, which is RET specific, and of course the RET gene is involved in those rearrangements. So it's an example of the reason that it's very important in my mind for us to urge our patients to have tumor tissue analyzed for these mutations, at least if they become resistant to radioactive iodine, and often even at the time of diagnosis, just to make sure that they don't have a tumor which is better suited to one of these agents than with radioactive iodine. So that's a summary of the state of play here in Australia in 2022. It's actually a pretty good story. I think we're very fortunate that we do have access to lenvatinib for patients with differentiated thyroid cancer. We're also very fortunate that Novartis, who make cabozantinib, or who market cabozantinib, are very good at making that available through a compassionate use program free of charge to patients. That's been a terrific advance from our patients' point of view. And we're also lucky that because momentum has not been lost in thyroid cancer clinical trials, that we still have trials open for patients with medullary thyroid carcinoma in Australia. And they are particularly the selpercatinib versus cabozantinib study, and hopefully soon the study of pralsetinib versus cabozantinib. Now, the final study that I'll mention to you is an Australian-based study. It's based out of Melbourne. It's called the iFirst study. And this is a study in which drugs are being used to take people with differentiated thyroid cancer who have radioactive iodine refractory disease and turn them back into patients who have radioactive iodine sensitive disease. So what this study involves is obviously taking people who have been on tyrosine kinase inhibitors before, or perhaps people who haven't been on tyrosine kinase inhibitors such as lenvatinib before, and then treating them over a period of a month with two drugs, dabrafenib and trametinib, which have been used for other cancers, which have been shown to be able to turn on the little sodium iodide pump on the surface of thyroid cancer cells. So these little pumps on the surface of thyroid cancer cells are also present on the surface of normal thyroid cells, so that all of us who've got a thyroid can concentrate iodine out of our diets and can convert that into thyroxine and T3. As I've said before, many patients with thyroid cancer, differentiated thyroid cancer, eventually become resistant to radioactive iodine, and they lose the function of that little sodium iodide pump on the surface of their thyroid cancer cells. What these two drugs can do is, in a certain percentage of patients, turn that back on again, and then make these patients amenable again to treatment with radioactive iodine. So this is an ambitious protocol. It's being done at multiple centres in Australia, including Melbourne, from where it's based, Sydney, Brisbane, Perth, and I'm pretty sure Adelaide is involved as well. But it's a rugged protocol. So people have an assessment of their radioactive iodine concentrating ability initially. Then they go on to medication, and they come, the dabrafenib and trametinib, and they have to come off thyroxine for a whole month. So during that whole month, they become quite hypothyroid, and then they have a further scan using I-124 at the end of that month to show whether they've been able to have their sodium iodide pump turned on. And if they do, then they're given a dose of radioactive iodine again. So if this works, and it has been tried to an extent at Sloan Kettering in New York and abandoned in the end because the drug that they were using to turn the sodium iodide pump on probably wasn't the best choice. But with these two other drugs, we are hopeful that we may be able to turn some of these radioactive iodine resistant patients into radioactive iodine sensitive patients. Now, that's available in Melbourne, Sydney, Brisbane, Adelaide, and Perth. And if patients have radioactive iodine refractory disease, they should certainly talk to their endocrinologist, their treating thyroid physician, and talk about getting access, or at least assessed for eligibility for that study, because it could become a game changer if we can turn radioactive iodine refractory people into radioactive iodine sensitive people again. So that is the final study that we have here in Australia. As I say to all of you on this bright sunny Sydney morning, I think it has been a really good year since I last spoke to you. And I think there have been quite a number of advances in the last 12 months that I think are being quickly swept into standard of care, standard treatment. And there are also a number of trials that are still open. And our goal, of course, by engaging through ITOG with the American Thyroid Association, is to make sure that Australia is not forgotten when it comes to setting up these clinical trials. And I think we've demonstrated, not just at our site in Sydney, but at sites in Melbourne and Brisbane particularly, that we are quite good at recruiting people to these studies, that the nurses and clinical trial people that we have involved in those studies are really very, very good, and that we can deliver these studies on time, and perhaps even at a lower cost than sometimes our colleagues in America can. So with that, I'll have to stop this morning, and I'm sorry for the interruption of my phone heating up. It's now cooled down, and look forward to perhaps talking to you all again in the future. Thank you.
Doctor, you have time for one question?
Yes, of course.
Question is, what is the best resource to find a clinical trial on your specific cancer and mutation?
Probably the best source is an NIH website, which lists all of these trials. I think it's NIH Clinical Trials or something like that it's called. It's the best list of those globally. And most of these clinical trials that are being undertaken in North America now, we do have access to here in Australia. So that's probably the best source.
Okay. That's all we have for tonight. Oh, one more question. How to determine the change from resistance to sensitive again tumor?
Yeah, look, that's a good question. So the way we're determining that in the I-FIRST study is using an isotope of iodine called I-124, which is a very high energy isotope, and one which enables us to very sensitively determine whether the drugs that we've used to treat people have successfully turned those little sodium iodide pumps back on. So that's how we determine that people have become resensitive to it. How do we determine in the first place that a person's become resistant to radioactive iodine? The answer to that is we give people a dose of radioactive iodine and we do scans, and if there's no uptake, then by definition, they're resistant. So that's how we work that out.
Okay, and I think that's all we have, and I'll let you all go enjoy your beautiful Saturday. And thanks for joining us this year, and hopefully we'll see you next time.
Okay, thanks, Matt. Thank you very much. Sorry about the technical difficulties.
No worries. Everybody have a good day. Thank you.
Okay, bye. Bye.